To contract the muscle fiber, you're going to need the interaction from chemical and cellular components, and it will result in the movement of the myofibrils, and the actin and myosin are actually going to slide past one another, and the effect of that is to shorten the sarcomeres. The fibers will shorten and pull on the attachment points. They will pull the insertion towards the origin. So first we'll look at the neuromuscular junction. It's a synapse. We're going to talk more about synapses in the next chapter when we talk about the nervous system. It's also called the myoneuronal junction, myo being muscle, neural being neuron. It's a place where an axon from a motor neuron and a skeletal muscle fiber interact. They do not physically touch each other. There's a gap between them, but they are very close. The skeletal muscle fibers contract only when stimulated from a motor neuron. So parts of that neuromuscular junction include the motor neuron itself, the motor end plate, the synaptic cleft, the synaptic vesicles, and we will talk about the neurotransmitters involved. For skeletal muscle, the neurotransmitter is always acetylcholine. Acetylcholine is going to be released from synaptic vessels in the neuron that's connecting to that particular muscle cell. That acetylcholine will then diffuse across the synaptic cleft and bind to receptors on the motor end plate, the receptive area of the muscle cell. That acetylcholine is going to cause changes in the permeability of the muscle's plasma membrane or sarcolemma, and what it's going to do is allow a rush of sodium to come in. That sodium is going to generate an electrical signal called an action potential, and that action potential is going to lead to the release of calcium from the sarcoplasmic reticulum, and it's that calcium that ultimately will drive the muscle contraction. Myasthenia gravis is an autoimmune disorder in which antibodies are produced that attack or bind to the acetylcholine receptors on skeletal muscle fibers, which are at that motor end plate in the neuromuscular junction. And for that reason, people with myasthenia gravis may have as little as 1/3 of the normal amount of acetylcholine receptors, and that can lead to widespread weakness and muscle fatigue. There are some treatments. There are drugs that inhibit the acetylcholinesterase, which breaks down acetylcholine after it's been released. Immunosuppression to slow the production of antibodies. And then there are also some biologicals that are basically antibodies against those antibodies, and removal of the antibodies during a flare-up with plasma exchange. So the excitation contraction events are coupled together. That's the connection between the muscle fiber stimulation and the muscle contraction. So when a muscle is relaxing, the calcium ions are stored within the sarcoplasmic reticulum. And troponin, tropomyosin complexes, proteins, cover the binding sites on actin filaments. When the muscle becomes stimulated, sodium rushes in. That causes an impulse for the sarcoplasmic reticulum to release calcium ions into the cytoplasm. Those calcium ions bind to troponin and change its shape, alter its shape. And the troponin is holding the tropomyosin in place, so when you change the shape of the troponin, you change the position of the tropomyosin, and that exposes the actin binding sites, and those myosin can bind to the actin and form the cross bridges. Whoops, thought we only had one. So let's see, let's start right here with a relaxed muscle. In a relaxed muscle on the myosin we have ADP, and the actin binding sites are covered by tropomyosin held in position by troponin. When you get electrical signal, calcium is released from the sarcoplasmic reticulum. Those binding sites open up. The myosin chains can then reach up and grab them. The myosin chains would then contract forced contraction, and ATP would then release the myosin from the actin, and the actin would go back to the cocked position and have ADP on it. This is the basis of the sliding filament model of muscle contraction. So when the sarcomere shortens, the thick and thin filaments slide past one another. H zones and the I bands narrow. The Z lines move closer together. When the Z lines move closer together, the muscle gets shorter. So the thick and thin filaments don't change length. The amount of overlap between them is going to decrease. So if we start down here at a resting muscle, this is going to increase. The Z lines are here. There's a lot of overlap, and then as we contract it, we bring this Z line this way and this Z line this way, because these fibers are all going to come right to the edge of the thick filaments. And that is called cross-bridge cycling. So the myosin head attaches to the actin binding site, forming a cross-bridge. Myosin cross-bridge pulls the thin filament towards the center of the sarcomere. ADP and phosphate are released from the myosin. A new ATP binds to the myosin, and that breaks the linkage between actin and the myosin cross-bridge, or breaking the cross-bridge. That leads to the splitting of ATP to ADP and phosphate, and putting the myosin back in its cocked original position. When neural stimulation of muscle fibers stop, there are enzymes such as acetylcholinesterase that rapidly decompose acetylcholine in the synapse. The muscle stops when acetylcholine is no longer stimulating those receptors. The stimulus to the sarcolemma and muscle fiber ceases, and calcium is actively pumped back into the sarcoplasmic reticulum. And the troponin-tropomyosin complex covers the binding sites on actin, so myosin and actin can't bind, and the muscle fiber then relaxes. The energy source from this is ATP ultimately, but that ATP that we have in the cell in reserve is a relatively small amount. Muscles do have the ability to have an enzyme that most cells do not, called creatinin phosphate, which is an enzyme that can put a third phosphate onto ADP and make it a high energy ATP, and then we fall back on cellular respiration, aerobic respiration, glycolysis, and the Krebs cycle to generate ATP to keep muscle contractions going. So the aerobic phase is just going to be covered by glycolysis. Remember, glycolysis occurs in cytoplasm and produces very little ATP. If we want to get maximum amount of ATP, we go to the mitochondria in the aerobic phase, in which the citric acid cycle, or you may recall it as the TCA cycle or Krebs cycle, are coupled with electron transport. That produces the most ATP. Muscles also have a protein in them called myoglobin, which acts like hemoglobin, so they have an extra store of oxygen in their muscles to help this aerobic respiration to occur. However, when you are resting or when you have gone beyond the ability to use your muscles, you get into an anaerobic lactic acid threshold. There's a shift in metabolism from aerobic to anaerobic. That's during strenuous muscle activity. The systems can't supply enough oxygen, so it goes to lactic acid fermentation, you'll recall from biology. And this means that you have to replace the oxygen that was on the myoglobin. Your book still refers to it as oxygen debt. Some of the newer texts are coming out and getting away from that term, oxygen debt, but it's really the same thing that it is really oxygen debt. The amount of oxygen needs to be replaced. The lactic acid needs to be converted to something else, and that allows muscles to then again go back to aerobic respiration. So muscle fatigue is the inability to contract the muscle And some common causes of that are decreased blood flow, ion imbalances, loss of desire to continue to exercise, accumulation of lactic acid. That's somewhat controversial. You have to go a long way to get to that point for fatigue. A cramp is a sustained involuntary muscle contraction that can be caused by changes in electrolyte concentrations in the extracellular fluids in the area. And then it's also important for you to know that one of the major products of muscle contractions is heat. It's a byproduct. Muscles are a major source of body heat. More than half the energy released in cellular respiration is heat. Less than half of that goes to ATP. And that helps to maintain body heat, and that blood will transport that heat throughout the body. Okay, we're going to stop there.